Okay, welcome everybody to Brain Science of Creativity. This is part 2. And my name is Brian Lawler. I'm Deputy Executive Director of the Global Brain Health Institute based here at Trinity College Dublin and Professor of Rhodri Psychiatry. And in this session we've got 4 fantastic speakers, and we're really going to be talking about circuitry, mechanisms and the relationship to consciousness and human development. So first of all, a great pleasure to introduce Lorina Naci, who's Assistant Professor in Psychology and also faculty at GBHI. And Lorina is going to speak to us about creativity and consciousness.
Great. I'm very delighted to be here. Shall I go ahead?
Yes, sir.
Okay, so I'm on the screen. Perfect. So hello everybody, delighted to be part of this Creative Brain Week event, and today I'll be talking about how creativity can help us understand consciousness, which is arguably the central quality of human life. Now if I can advance the slides, I will tell you even more about that, but maybe I can do it this way. Just a second. There we are. So the stories we tell are a paradigmatic example of creativity, and the stories that we tell ourselves and tell others are basically shape the way that we understand the world. For example, movies by masterful directors such as Alfred Hitchcock entrance us. They create a shared experience between us that we can enjoy and relate to one another with. And in my work, I have shown that there is a neural basis for this shared conscious experience. So here on the screen, I'm showing the brains of different individuals, and I've shown that the frontoparietal network, shown there in red, bilaterally across both hemispheres of the brain, is exhibiting activity that is ticking along with the same ebb and flow in different participants when we are engaged in a highly interesting narrative. And indeed, it is as if the brains of different people are synchronized. So I hope that throughout my presentation, you will be highly synchronized to one another, that you'll find it very interesting. But that is the idea. That is how we experience the stories and the narratives that really leave a mark and impress us. And by different experiments in groups of healthy individuals, I have shown that both the way that we pay attention, so the cognitive demands of a narrative, how it is taxing our attention, higher level cognition, as well as our emotional perception. For example, in dramatic Hitchcock story, the way that we are feeling suspense throughout the movie on a moment-to-moment basis. Here I used a movie by Alfred Hitchcock where a young boy takes his uncle's revolver and is not aware of his power and danger, and is waving it in public and threatening bystanders. It's a kind of edge of a seat short narrative, and our perception of suspense, as well as how attentively demanding a strong narrative like that is all realized through the workings of this frontoparietal network of the brain, which I've shown here in different colors, and that is actually a correlate of conscious processing. So if we put healthy individuals inside the neuroimaging scanner and measure their brain activity, which I have done while they are deeply anesthetized with propofol, which is the most common clinical anesthetic agent, the brain activity of the frontoparietal cortex is abolished because individuals are not consciously aware. So this work has established that, again, the frontoparietal activity, the similarity of this network over time in different people is the neural basis of that common conscious experience. And with this, then, we can establish a neural signature, which I've illustrated here in red, of the narrative understanding that can be dissociated from other sensory processes such as audition, shown in yellow, and vision in blue. And that can help us measure conscious awareness when the behavioral manifestations of it are missing. And so this is not only a intellectual conundrum, but also it's very important for understanding consciousness in individuals that through different conditions such as severe brain injury become unable to speak or willfully express themselves, and therefore are sometimes deemed to be unconscious mistakenly. So although consciousness is a shared attribute to all of us, it's the central quality of human life, we are not private to the conscious experiences of other people. We rely on them telling us what they think and feel, and when those indicators are missing, then we are left in the dark with regards to whether they do indeed experience the world in the same way as we do, or whether they are conscious at all. And so in the next slides, I'll show you how we can use this neuroimaging approach to investigate consciousness when it is at question. So due to improvements in intensive care, technology, a significant and increasingly number of patients who survive brain injury progress to the states where they are behaviorally non-responsive, so they do not respond willfully to commands delivered by the clinical staff at their bedside and in repeated number of times. And on this basis, they are thought to lack consciousness and are diagnosed to be in a vegetative state. And I have used this work that I have just described to test a number of patients, and I will discuss here 2 patients who are very similar clinically and behaviorally, and that are thought to lack consciousness, met all the internationally agreed upon criteria of the vegetative state for several years. So a female who's 20-year-old and had an etiology or is unknown why she became unconscious in the first place. I saw her when she was diagnosed as unconscious for 6.5 years, and a male who, 34 years old, whose brain was deprived of oxygen for a period of time, became unconscious, and then for the next 16 years was repeatedly diagnosed clinically to be unconscious. And when they saw the movie that I've just described to you inside the scanner, we found something striking. So I'm going to just very briefly guide you through this slide. And basically, in the top row, we have the healthy group with the responses, auditory responses, visual responses in pink and blue in the healthy individuals, and the frontoparietal responses in different colors, tracking different aspects of the movie that lead to its understanding. Patient 1 had highly synchronized activity in auditory cortex, but nothing in visual or frontoparietal regions, suggesting that in the best case scenario, this patient did not process the narrative, did not understand for whatever reason, maybe not even related to unconsciousness. We remain agnostic to the true state of this individual, given that they cannot move or speak willfully. But when we look at the second patient, we saw very striking that despite the diagnosis of unconscious for 16 years, we have very strong activity that in auditory cortex that highly synchronized to healthy individuals as well as in the visual cortex, but critically activity in the patient's frontoparietal cortex was highly synchronized to that of every healthy participant, and the perception of suspense and emotion over time, suggesting that this individual not only was conscious, but indeed could understand narratives and follow them as healthy individuals over time. And this work established that we can detect covert awareness in non-responsive individuals, and that by covert narrative capacity, we mean here the ability to understand narratives over time, so highly preserved levels of conscious awareness. And the patient that I've just described is called Jeff. Here's portrayed with his father, Paul. I learned after the study that the father, Paul, would take Jeff to the movies, had done so for 10 years on a weekly basis, taking Jeff out of his long-term care facility. And this was something that they enjoyed doing together, and it was a way for Paul to feel connected to his son. And he was extremely happy to hear my results, and said to me that all those times he spoke to Jeff in private, now he knows that he understood him. Another study that I want to very briefly touch upon is how we can then probe conscious awareness, hidden awareness in individuals who have their eyes closed and cannot follow movies. So for this, I developed a similar approach, but with an auditory-only narrative, which is here. You might have even seen the movie Taken. It's not the best, work of art, but in it there is a scene, a 5-minute piece that's very dramatic, where the father is speaking to the daughter on the phone while she's being abducted, and we can then characterize the meaning and the attention demands of the story. And when we do so with inviting healthy individuals in the laboratory and telling them how they rate the suspense over time, you can see in different colors here, we can see their rating of suspense is very similar, even though they look a little bit noisy. Actually, that's quite similar, with the suspense ramping up when the daughter is being abducted here at the end, indicating the average in red. And then when I do semantic analysis of the story, we can very closely link the perception of suspense with the plot development. So naturally, suspense is an indicator of understanding over the time course of a narrative. And then we can see that in the healthy individuals, the frontoparietal activity is very synchronized over time to the story, which you can then use to understand patients who don't have their eyes open but are in this very non-responsive state. So I'll talk here about a 19-year-old male whose brain was deprived of oxygen for a period of time. And when I saw him, he was only 2.5 months after the injury and was non-responsive, diagnosed to be in a vegetative state. And here is his response to the narrative. On the top here is the healthy group with the auditory and no visual activity, obviously, and the frontoparietal synchronized activity over time. And the patient equally had highly synchronized activity in the auditory cortex as well as in the frontoparietal regions, suggesting that this patient was not only consciously aware, but also understood this and felt suspense throughout this story in the same way as healthy individuals. So again, very surprising finding. But what happened was that 3 months after I scanned this patient, he actually recovered consciousness. He recovered talking, he recovered feeding and standing with assistance. And I was very keen to find out what he actually thought, what his experience was when he was in a state clinically diagnosed as vegetative. So I invited him back to the laboratory and performed a memory task, a forced choice recognition task, presenting stimuli that he had seen before and stimuli he hadn't. And what I found was that Paul, no, this is Huang actually, that the patient did remember the objects that he had seen during his visit. He also spontaneously recalled the names of some of the research staff that he had met and the details of the movie, the story that he had been presented with while in the fMRI scanner. So this very striking case of preserved memory during the presumed vegetative state, again suggesting that in some cases consciousness is preserved at very high levels, and this has important clinical implications. Moving right along, I want to give you another example of how we can understand consciousness in states where willful behavior is suppressed, such as during general anesthesia. Between 0.2 to 5%, and individuals who undergo general anesthesia have what is known as accidental awareness under anesthesia. And this is a very traumatic experience for the majority of the individuals who experience it. Up to 71% do experience post-traumatic stress disorder and other mental health problems related to it. The true incidence is impossible to ascertain with current methods because it is assessed with the post-anesthesia structured interview, but the anesthetic cocktails that we would receive if we have general anesthesia induces amnesic effect. So most individuals don't actually remember their experience. However, studies show that even for those individuals who have no memory of the full experience, there is negative or psychological harm related to experiences of awareness under anesthesia. So to address this question of the extent of preserved consciousness and processing during anesthesia rather than after it, I developed this, designed this study, which has been published in several papers. It's a very unique way to actually look at what people are thinking about when they are unconscious inside the scanner, generally anesthetized. And what I did was anesthetize individuals with a clinical team up to deep sedation, level of general anesthesia, at which point they're considered to be clinically unconscious. And listening to the same story that I have just described. Now, as I mentioned, when we look at the whole group together, we see that when they are awake, they have very robust response from the parietal network to the narrative, and naturally this is abolished when people are deeply anesthetized, and this is all well and good when we look at all the individuals together. But what we can do with this approach that I have developed is look at every individual separately. And when we do that, we strikingly find that in 2 participants, so 12% of the group, had highly synchronized activity in their frontoparietal cortex. So we're talking number 4 and I think is 16, I can't see there. But these 2 individuals, bilaterally, frontoparietal cortex highly synchronized with the cognitive demands of this narrative, suggesting that they were consciously aware, despite those clinical indications and designation of unconsciousness. And finally, I want to tell you very briefly about sleep. So sleep is obviously a wonderful thing, and most of us should get more of it, apparently, research suggests. But sleep is not a monolithic phenomenon. We go through different sleep stages, and with earlier stages actually are not shown here, but earlier, even before non-REM sleep, being lighter, and then as we follow deeper into sleep, the idea is that we are unconscious of the environment and we process little to next to nothing of what's happening around us. However, really the extent to which we process the environment when we are in sleep has been largely unexplored scientifically. And we are starting to do so now with the methods that I have developed. And what I saw when individuals went to the scanner and fell into night sleep, that went through the different stages of sleep while listening to the narrative I just described, what I found was that you have 5 participants here at different stages of sleep. And 4 out of 5, but regardless, people at different stages of sleep can process auditory information from their environment. So low-level sensory information that is processed in the brain as noise or perhaps stimulation that's not meaningful is processed throughout sleep. So that is the first thing that we found, and this is perhaps not the most novel thing, but is the first piece. And the second piece is that one individual who was in REM sleep, so to current state of understanding, not aware of their environment, had highly synchronized activity in their frontoparietal cortex bilateral to the cognitive demand and the semantic requirements of this narrative, again suggesting that this individual understood and meaningfully tracked the story while they were in REM sleep. So I've gone very briefly through different studies just to give you a taste of how narratives can help us to understand shared conscious experiences. And the highlights are that my work has established that strong synchronized activity in the frontoparietal network of the brain is the basis for our shared conscious experiences during engaging narratives, and more broadly, during engaging experiences that cause this shared conscious experiences. But within the context of the narrative, then we can look at this neural signature of understanding, which is very useful for helping to reveal hidden consciousness in a number of groups, and that in particular can also tap very highly preserved levels of consciousness that we would otherwise have no access to. And this creates a potential clinical marker for improved detection of consciousness, and also it can help us understand the expression of consciousness in altered states of being. And I want to thank you very much for your attention, to thank my students, collaborators, funding bodies, and the research participants. Thank you again.
Thanks, Lorina. So our next speaker, it's a recorded session, which we're going to hear is from Dr. Dariazabelina. She's assistant professor at the University of Arkansas in the Department of Psychology, and she's also director of the Mechanisms of Creative Cognition and Attention Lab. So we're going to hear about the mechanisms underpinning creativity.
Hi everyone, my name is Dariazabelina and I'm joining you today from the University of Arkansas in the United States. I'm happy to be here for the Creative Brain Week and to share my work on examining creative cognition with EEG. But first I'd like to take just a few moments and motivate the study of creativity. Those of you who are familiar with TED Talks might know that the #1 most viewed TED Talk is by the now late Sir Ken Robinson and is titled Do Schools Kill Creativity? So creativity in general is an important topic, it's popular, it's something that the public is generally quite interested in. But really, if you think about it, children who started school this year will be retiring in 2086. We don't know what the world will be like then, we don't know what the world will be like in 5 years or even tomorrow. So as the old adage goes, the only constant is change. And what I would like to suggest is that what we should be educating our children is for independent thinking, adaptive problem solving, success when meeting new and unexpected challenges. So really we should be educating them to think creatively. However, if you spent any time outside lately or on the playground or on college campus, this is the sight that you probably have encountered, at least at some point. Our children and even ourselves, we spend a lot of time looking at our devices, smartphones, watching TV, playing video games, generally spending little time outside. And what this is all leading to is fewer chances to engage one of the networks that is really important for creativity, and it's called the default mode network. Just as a quick overview, our brain is comprised of 6 are displayed here, but 7 different networks that might be activated when we're engaged in certain cognitive processes. And there are 2 networks that are especially, that we know from the literature now, that are especially important for creative thinking. The first, as I mentioned, is the default mode network. And generally what we know is that it is activated, it's a set of brain regions that are co-activated when we're internally focused, when our attention is directed inwards, and when we're engaged in self-generated cognition. Another network that is known to be also quite important for creative thinking is the control network, or it's also called the frontoparietal control network. And also from the literature, we know that it is generally important for error monitoring, initiation of the task, it's a core network for task control. So both of these networks are really quite important for creative cognition. So before I go more into the neuroscience of creativity and share some of the neuroscience work from our lab, I would like to also discuss what we mean by creativity and how we measure it, particularly in our laboratory. One of the tests that we used are laboratory tests of creativity. These are sometimes called diversion thinking tests or alternative uses tasks. They're very well known, very well established tasks of creative thinking. These tasks measure the ability to think in novel and flexible ways. And one of the sort of common instructions for participants is to name as many creative uses for a common object, such as a brick, and participants are usually given a limited amount of time, such as 2 or 3 minutes, to generate novel uses. And responses are generally scored for fluency, which is the number of ideas that people come up with within that limited amount of time, and also originality, or that is how unique or original the responses are based on a sample or some types of standardized norms that exist. So in addition to these laboratory tasks, we also have used, we have asked people about their real life creative achievements. So instead of just asking them to produce some novel uses for a common object in a laboratory within a very limited amount of time, we try to approximate real life creativity by using the Creative Achievement Questionnaire that asks participants about their achievements in the real world within 10 creative domains such as music, visual arts, but also engineering, scientific achievements, and so forth. So based on a number of studies using behavioral, EEG, genetics, and fMRI techniques, I have designed a model of creativity and attention that sort of highlights that participants or people who are particularly good at performing on laboratory tasks of creativity, such as divergent thinking and alternative uses tasks. They also show elevated cognitive control and flexible attention, as well as selective sensory filter. However, people who have long-term creativity, so people who have higher number of real-world creative achievements, they also typically tend to have higher levels of cognitive control, but interestingly, they might also have more diffused attention and diffused sensory filter, and I'm going to share in just a minute what I mean by this. Okay, so one study that this model is based on examined how divergent thinking relates to cognitive control. In the study, we looked at the N2 event-related potential, or ERP, which is a standard marker of cognitive control. And what we found was that based on both behavioral and ERP data, we found that indeed divergent thinking relates to higher levels of cognitive control. So people who perform better on divergent thinking tests, those laboratory tests that are timed, where people are given 2 or 3 minutes to come up with creative uses for a common object, people with better performance on those tests also had better cognitive control. In another study, we also looked at creativity and sensory processing. Here we looked at the P50 ERP. So this is a marker of sensory gating or filtering of sensory information. And 50, by the way, means that it's we look at the brain response only 50 milliseconds after stimulus onset. So this is very, very early, almost brainstem response to stimuli. In this particular task, people are given they hear 2 auditory clicks or pairs of auditory clicks, and they are asked to just listen to them. They're not asked to respond, and we're just measuring their brain responses or brain potentials to each auditory clicks. By the way, they're separated only by 500 milliseconds, and they hear quite a few of these pairs. So interestingly, what we find is that people with real-life creativity show what we call leaky or more diffuse sensory filter. So they are not as, they do not attenuate that second click compared to the first one. They don't show as much habituation. So we might say that they are indeed a little more open-minded or open-brained. However, people who perform well on laboratory tests of divergent thinking do show more selective sensory filter. They do attenuate that second click quite well. They do show very good habituation on this particular task. And these 2 effects, by the way, are reliably different from each other. So that's interesting that the effects are quite strong. Okay, and so finally, in one more study that I'd like to share from our lab, we looked at alpha band. Some of you might know that alpha activity in the brain is generally higher when we close our eyes, when we're relaxed, when we are internally focused. You know, you might think of when you're kind of on the bus looking out or on the train and you are daydreaming about something when you're not really externally focused as much. So we looked at this type of brain activity and involved machine learning while examining divergent thinking. On this task, participants were given the following task, or this was the structure of the trials. They first saw a fixation cross, and then they were given a cue, either an N that stood for normal or a U that stood for unusual, meaning that on this particular trial, they were to generate either a normal or an unusual use for a object that they saw. so for a brick in this case, and then they would type out that response. And we looked at their brain activity while they were generated either novel versus compared to the normal compared to the unusual uses for objects. As you might expect, participants took a lot longer to generate unusual compared to normal uses. That's in terms of the reaction times. Interestingly, but also in line with a lot of previous studies, we found that there was greater alpha power, so greater alpha activity when people were generating uncommon compared to the common items, particularly in the right frontal and right parietal regions of the brain. But what was really novel in the study and really quite interesting is that we were able to use machine learning to classify high versus low creative people. So depending on whether they were generating interesting, high, novel, unusual uses compared to the more common uses, we were able to classify these individuals with 82% classification accuracy, whether they were more versus less creative. As you can see, we can learn a lot from using neuroscience techniques, particularly in our case EEG techniques, to understand creative cognition, something that I'm excited to keep doing in the future. I'd like to thank everyone in my lab, the MoCCA lab, my collaborators, sources of funding, and thanks so much for your attention. Please do reach out if you have any questions.
Great. So we'll move quickly on now to the next talk, which is by Rhodri Beaty. He's Associate Professor of Psychology at Penn State, and he's also Director of the Cognitive Neuroscience of Creativity Lab. So I think this talk follows very nicely on Dariazabelina's talk. And this is also pre-recorded.
The meeting so far. My name is Rhodri Beaty. I'm an Assistant Professor of Psychology at Penn State University. And I'd like to talk to you today about some of my research on the neuroscience of creative thinking. Okay, so I'll just start off with some of the big questions that have motivated my research over the years. And perhaps the biggest question of them all is, how does the brain produce creative thought? Where do creative ideas come from? How does the brain flexibly piece together information to help us solve new problems? And to what extent does creativity rely on memory? So memory obviously provides a foundation for creative thinking, but are there some cases where memory can kind of get in the way and prevent us from seeing something in a new light? And to what extent is the creative brain wired differently? And so this means, from an individual differences perspective, are there patterns of neural wiring in the brain that differentiate more creative individuals from less creative individuals? So with some background in the creativity literature, creative thinking is often defined as the ability to produce something that is both new and useful. And often studies will use tests of divergent thinking to measure people's ability to think creatively. And the nice thing about divergent thinking tasks is that they tend to correlate with real-world creative behavior and achievement. So people who do better on these tasks also typically report having more creative hobbies and activities and achievements outside the lab. So more on divergent thinking. So as I mentioned, divergent thinking is the ability to generate different ideas. So it's the ability also to generate ideas that specifically diverge from a given concept or starting point. And so researchers will measure divergent thinking with both verbal-based tasks that involve words and word associations, and also drawing-based tasks that present incomplete shapes and ask people to make creative drawings. I'm going to focus more on the verbal tasks today. And here's an example of someone who had completed a drawing task, and you can kind of see kind of where they go with these different starting points. And the thing about divergent thinking is that it's thought to differ from convergent thinking in that there's no correct solution. So with divergent thinking, you're just coming up with multiple different ideas, and there's no single right answer. Convergent thinking, you're trying to converge on a single correct answer. So there have been a number of theories over the years that have been proposed to attempt to explain how people come up with creative ideas. And one theory is known as the associative theory. And according to associative theory, creative thinking involves spontaneously connecting remote concepts in semantic memory. So making connections between concepts that are kind of far apart. And these concepts are stored in the semantic system of the brain, so semantic memory, which is the system that represents facts and concepts, and they're thought to be organized in interconnected networks. So things that are more related or associated with each other tend to be closer together in these networks. And according to associative theory, individual creative ability relates to the strength of associations in memory. So people that have really strong associations between common words will tend to be less creative, and those who have weaker associations to those common words will tend to be more creative. Okay, and more recently, there has been another theory that has been proposed to try to explain how people come up with creative ideas, and it's known as the executive theory. And according to executive theory, creative thinking involves cognitive control and executive functions. And so executive functions help us to regulate our thoughts, emotions, and behaviors. And on the right-hand side here, you can just see a few examples of different executive functions, things like controlling attention, working memory capacity, cognitive flexibility, inhibition. So basically the ability to exert some level of control over the direction and contents of our thoughts. And so this ability to control cognition and attention may help us to manage the interference coming from common and unoriginal ideas when we're trying to think creatively. So when you're trying to think creatively, you want to think of something new, often something that you haven't thought of before. And so often in that process, things that we already know pop into mind. And what cognitive control and these executive functions might do is help to kind of block those things from disrupting the creative thought process and shifting us more towards fruitful lines of thought. And so even more recently, there's been attempts to kind of combine the associative and control attentions or executive theories. And this is a perspective that says that creative thinking involves both associative and executive processes. So not just one or the other, but some combination of the two. And we did a study several years ago, a behavioral study, looking at the contributions of these different cognitive abilities in predicting people's ability to think divergently. And we found that both abilities were predictive of divergent thinking. So it wasn't just like one was predictive over the other, they both made unique contributions to divergent thinking performance. And that raised the question in my mind about kind of how this works in the brain. So we had some behavioral evidence with correlations and tasks that people in the lab. We actually wanted to know more about what's playing out in the brain when people are trying to think creatively. And over the years, I've been developing a framework that helps to explain the neuroscience of creative thinking. And it focuses on the brain's networks. And so when I talk about networks, I'm talking about several different regions across the brain that are working at the same time. So not just a classic fMRI study where one area is lighting up or another area is lighting up. Networks, or network neuroscience, really is seeking to understand how regions communicate across the brain. And so there's a few different networks that have been associated with creative thinking over the years. One is called the default network or the default mode network. And this got its name because it was showing increased activity kind of at default baseline when people are in an fMRI scanner and not doing anything in particular. This network was kind of lighting up, so to speak. And the default network has been associated with many different aspects of thinking, things like mind wandering, daydreaming, memory retrieval, imagining a future experience, a lot of things that involve kind of inward thinking that's memory-based or imagination-based. And in terms of possible creative functions that the default network may be serving, could be things like idea generation or mental simulation, so kind of like brainstorming possibilities. Another network that's been associated with creative thinking over the years is called the executive control or the frontal parietal network. And this network plays a general role in things like cognitive control and response inhibition. So again, those executive functions that I was just mentioning. And in terms of possible creative functions, the executive network may be playing a role in more strategic or controlled aspects of thinking that involve things like idea evaluation. So once you've already generated an idea, this network may be involved in evaluating it for how it might work. And also things like inhibiting common ideas. So when common ideas come to mind, we want to kind of move away from them. So this network might help us to do that. A third network that's been implicated in the neuroscience of creativity literature over the years is called the salience network. And the salience network plays a general role in what's called salience detection, so picking up on important information in the environment or internally, and also just kind of plays a general role in switching between the first two networks I talked about, the default and executive. And so the salience network may play a role in terms of creative functions in identifying candidate ideas that may come to mind from the default network, and then kind of switching over to a more evaluative mode of thinking with the executive network. So kind of maybe switching between a generative mode of thinking and a more evaluative or controlled mode of thinking. An interesting thing about the networks that I've talked about so far, specifically the default and executive networks, is that they have been shown for a while to not really be working at the same time. So when one network activates, the other network tends to deactivate. And this kind of makes sense when you think about what they're kind of doing generally for cognition. So for example, if we're mind wandering, then the default network's activated. We don't need to be kind of exerting some level of control over our thoughts. And conversely, when we are trying to exert some level of control over our thoughts and focus our attention on a demanding task or problem, we want the control network to be activated and not so much the default network that might be kind of interjecting mind wandering thoughts at that time. And so these networks kind of have this antagonistic relationship that has been identified and shown several years ago. But I noticed in the creativity literature that many studies showed activation of regions within these two networks. So not connectivity, but activation. So one area was lighting up, another area was lighting up, but they were within these networks that typically don't work together. And so that raised the question in my mind as to how they might be working together when people are thinking creatively. Okay, so I did a study back in 2015 with some collaborators where we put people in an fMRI scanner and asked them to think of creative uses for objects. So this is kind of a classic task of divergent thinking. And so first we identified a whole brain network associated with divergent thinking. So these are just some regions in the brain that were connecting to some other regions when people were thinking divergently. And then we focused in on regions of interest. So in this case, the posterior cingulate of the default network that I mentioned earlier. And we looked at what other areas of the brain that region was connecting to or communicating with. And we found that the posterior cingulate was more functionally connected to frontal areas within the executive and salience network, so dorsolateral prefrontal cortex or anterior insula. And we also looked at this connectivity pattern across time. So when people were first trying to come up with ideas at the beginning of each trial versus at the end of the trial before they kind of told us what their idea was. And here we found that the posterior cingulate was showing increased functional connectivity with the insula of the salience network at the beginning of the task, and then later connectivity with frontal areas and other regions. And the way we interpreted this is that at the beginning of the task, people are maybe in a generative state, which is a kind of default network role, and the default network was coupling with anterior insula that's involved in switching. So the insula is maybe kind of grabbing a promising idea and then passing it along to frontal areas for further evaluation. And so shortly after we had published these results, we published a review paper summarizing some of the findings that we have been seeing in the literature in terms of artistic creativity. And so what this research was showing was that not just divergent thinking, but also specific creative domains like musical improvisation and artistic drawing were also showing functional connectivity between the default and executive control networks. And what these studies also showed that we didn't show at the time was that idea generation and evaluation were showing different connectivity patterns. So we were just kind of venturing what the cognitive processes were in divergent thinking, but these studies were actually showing that different connectivity patterns were emerging as a function of different creative thought processes, with more connectivity between the networks found during the evaluation stage. And what this suggests to us and to others at the time is that network coupling may depend on the goal-directedness and constraints on thinking. So even with music improvisation, for example, which is thought to be kind of a characteristic, spontaneous process, researchers found that the default network was correlated with or connected to the control network, specifically when musicians were trying to improvise based on a specific emotion. And so the way this was interpreted is that musicians kind of have to keep the emotion in mind. So say you want your performance to express joy or sadness or some other emotion, you kind of have to keep that in mind while you're spontaneously generating ideas. So it's kind of this idea of keeping a goal active in mind. So the control network may be kind of guiding what the default network comes up with to kind of meet the goals of the task, which in this case was thinking or improvising based on a specific emotion. And another study in artistic drawing found the same connectivity pattern, but specifically when artists were evaluating ideas that they had previously generated. So again, more kind of controlled aspects of thinking are associated with default and control network functional connectivity. And so to kind of go back a few slides to where I talked about that fMRI study that we did, we actually did also an exploratory study where we looked at individual differences in divergent thinking. So how people varied in their performance to think divergently. And we found that people that showed greater global efficiency between regions within these three networks I've been talking about so far, default, salience, and executive, tended to produce more original ideas. So there was a kind of a tentative preliminary correlation between information processing within this system and also the quality of ideas people came up with. And so that raised the question at the time of whether the creative brain is wired differently. And so at least in the context of divergent thinking, if there's a pattern of brain connectivity that are helping, that's helping people to think creatively, and that varies across people. And so we did a follow-up study where we scanned many more people. We scanned about 163 people, and doing the same divergent thinking tasks where we're in the scanner thinking of creative uses for objects. And we tried to find a whole brain network that was predictive of creative thinking ability. So we used a machine learning approach, and we searched throughout the whole brain, and we tried to find brain regions and connections that were predictive of people's ability to think creatively. And we found what we called a high creative network, and this was just connections that were predictive of creative thinking ability in this study. And out of the 35,000 possible connections that were in the network that we investigated, the most strongly connected regions and connections were within the default, salience, and executive regions. And we were able to predict a person's divergent thinking ability just based on the strength of the connectivity between these regions. And importantly, this finding in the original sample also extended to independent samples from different countries. So we identified this network pattern and applied that over to these other data sets where people were doing divergent thinking in fMRI. And also just resting in the scanner, we found that those with stronger connections between regions within these three networks tended to produce more original ideas. Okay, so just to kind of wrap up and summarize, what we were showing in our research is that the default, salience, and control networks dynamically interact to support creative thought, and that these networks appear to support both general creative thinking and creativity in specific domains. And as I just mentioned, in terms of individual differences, people with stronger connections among these networks tend to produce more original ideas. So in terms of other ongoing and future directions I didn't have time to talk about today, we're also interested in understanding more about the cognitive mechanisms of these brain network interactions. So not just showing that some network pattern is associated with creative thinking ability, but trying to find more about what are the specific cognitive processes involved. And so several studies have found that the interactions between these networks are associated with things like goal-directed memory retrieval or inhibiting responses, and also kind of focusing attention inward. So we're interested in understanding more about specific cognitive processes that are tied to these interactions of these rather large networks. And also we're interested in studying the network neuroscience of creative thinking longitudinally. So in the studies I talked about today, we just scanned people at a single time point and correlated their brain connectivity with their creative thinking scores. And so in the future, we kind of want to see how these networks might change gradually over time, across development, across the lifespan. We also are doing some research in older adulthood and seeing how these networks interact in individuals as they age. And also if there's ways to intervene and to strengthen the connections between these networks to then kind of improve people's ability to think creatively. Okay, that's all I have for today. Thank you for your time and attention. I just want to briefly thank my sponsors and funding agencies, National Science Foundation and John Templeton Foundation. And I hope you all enjoy the rest of the event. Thank you.
So now we will move to our final speaker in the session, Professor Rhodri Cusack. He's the Scientific Director of TCIN, Trinity College Institute of Neuroscience, and he's the Thomas Mitchell Professor of Neuroscience. And he's going to talk about, I guess, creativity. It's going to be child brain and creativity, and perhaps creativity across the life cycle.
Indeed. So thank you very much for the invitation, and thank you all for coming today. So I'm going to tell you in particular about the brain systems, what we're learning about the infant brain. So the goal of my group is to understand what's happening in infants' minds. There are lots of ways we can do this. One of them is we can give infants tasks to do, and we can try to watch their responses. This is obviously, there's been tremendous progress made with this method, but it's very hard. Infants, we're talking about pre-verbal infants in the first year. We have to carefully watch their responses. And sometimes they just are not revealing what's going on the inside. So our approach is to use MRI with young infants. So this is our son, Dylan, going into the MRI scanner here. So the fantastic thing about MRI, as you've been seeing today, is you can use it to measure lots of different things about the brain. So you can measure not just the structure of the brain, but the activity in the brain and the connections in different parts of the brain. There's been quite a lot of work. Again, as you've been seeing so far, I've had some great sort of introductions to this. There's been a lot of work using fMRI in adults. And one of the things we've learned is that there is various different networks. And these range from, for example, the sensory systems, the networks that kind of receive the information from vision, from audition, from touch, and then right up to much more complicated, higher cognitive networks. And these are brought online when you're doing things like solving a difficult problem, or when you're learning a new skill. If you're trying to learn to tango, those initial lessons where you're having to think through every single thing that you can do before you're being able to sort of backsource, automate stuff. That moment when you're thinking everything through, that's what your executive control network, that's what these higher cognitive networks. I'm going to focus on it's called the executive control network sometimes. I'm going to call it the multiple demand network. It's another name for the same thing. It's also, we've learned, very strongly involved in creative thinking. So that, for example, the alternative uses task here, thinking of all the things you can do with a brick. So what we're going to do is we're going to look in infants. The approach here is to look in infants and see how mature some of these networks are. And in particular, this higher cognitive network, we're going to focus on the multiple demand network. What do we know about this already? Well, there are several different bits of evidence. I'm just going to tell you about one of them. So the synapse is the point you probably know is the point where one neuron, it's a little gap between two neurons, it's when one neuron is passing information to the next neuron. And so people have looked at synaptic development at the beginning of development through infancy and childhood. And what they found there is that earlier networks, sensory networks, the synapses are developing before. All the synapses kind of go up and then they get pruned away. So you can see these kind of bumps. The sensory networks develop first, then sort of higher level networks like language networks, and finally these very high cognitive networks. So according to this view, infants will be showing fairly perhaps basic, they won't have this higher cognitive function, they'll just have this sensory stuff. However, if you look at these curves a little more closely, even by 2 months, the prefrontal synaptic density is at a quarter of the level of its peak, and it's already higher than the level that you have in adults, sorry. So potentially, the question here is this prefrontal glass, is it half full or is it half empty? Now, from behavior, I mentioned we have also used behavior. Now, infants don't do well at the sorts of tasks we use to measure prefrontal function in children or in adults. If you ask them to solve that puzzle or learn to tango or to come up with all the creative uses of a brick, they're terrible at all of those things. So according to that view, maybe not much going on there. We also know that they're formidable statistical learners. So it's been shown, if you show sequences, like these little sequences of monsters here, if you structure it so that they come in little triplets, the green monster always follows the yellow one, the purple one always follows the green one, but these triplets can be in any order. It turns out that after just a short period of exposure, infants will start to learn those triplets. They will recognize the triplets, and if you violate the triplets, they'll look surprised. Now, all of this has led to the common view that infant learning is a kind of passive, bottom-up process, that, maybe they're looking out for patterns, they'll start, and you've got this sort of sequence of maturation. You start off looking for patterns in your primary your primary sensory cortex, and then perhaps the secondary cortex will start learning the patterns from the primary region, and then your higher regions will learn from those other ones. And it's a sort of sequential progression. However, against this is a rather interesting observation, and this is that infants can actually learn quite abstract rules. They can learn, for example, to pick out the patterns of 2 things repeating and then one being different. They can learn that, and they can abstract it to a new situation. And this is a hint that perhaps there's something more complex going on here, that they're not just passive pattern recognizers. So our plan is I'm going to give you a few different examples, just quickly, a few different examples of how we're looking at this executive control network using functional MRI, using brain scanning. So the first one, you've heard of this method a little bit today. I'm just going to unpack it a little bit for you. This is what's called resting state fMRI, and this is a classic way to look for these brain networks. So what you do is you measure the brain activity through time. So the baby will be in the scanner maybe for 5 or 10 minutes. We'll look at their brain activity. You want to use this method, you can just leave them resting in the scanner. They can even be asleep. What you find when people are resting in the scanner is that particular bits of the brain, the activity will be fluctuating in time. It fluctuates quite a lot. What we can then do is we can measure all the other bits of the brain. So here I'm just showing you, the square and the triangle as well, and these regions will all have their own time courses. What you can then do is you can say, well, how similar is the pattern of fluctuations in this bit of the brain to this other bit of the brain? And if they're very, very similar, if they're fluctuating in the same way, they must be somehow connected together. They're sharing information. So we can visualize that. Here I'm showing this orange line. The warmer colors are showing you a stronger connection because the circle and the triangle have more similar patterns of activity, whereas this square is quite different. So using this method, we can start to piece together the networks of the brain. Okay, so let's get down to business, and let's look at it in the multiple demand network. So this is a kind of 3-dimensional rendering here in an adult, and the warmer colors here showing you stronger connectivity, and the cooler colors showing you weaker connectivity. This is within this multiple demand or executive control network. Let's look down in infants, 3 and 9 months. When we first ran this analysis, we completely expected, the reason we did 3 and 9 months, we thought 3 months is going to be nothing. 9 months is going to perhaps building up a little bit on its way. But what we found was that actually many characteristics of this adult network were already present even at 3 months. So you can kind of see it in these 3-dimensional renderings here. You can also see it if I show you the pairwise connectivity between every region and every other region. Or we can do something called hierarchical clustering, which kind of looks for the common bigger structure within this network. So you can see that the 3-month structure of this network and the adult are incredibly similar. So this executive control network appears to be really surprisingly mature, even right down at 3 months. Now, so this multiple demand executive control network is potentially already kind of set up in some way. But unless it's connected to the rest of the brain, unless stuff can find its way there, it's not going to be any use. So there's a theory out there that long-range connectivity in the brain actually lags behind short-range connections, so that what happens during development is initially all these local regions are connected together, and through development you start connecting together regions that are far apart. And this work used resting state fMRI, and it goes back a little bit now. However, there are some significant concerns have developed around those experiments, and in particular, it turns out when you're trying to measure connectivity of brain regions that are a long way apart, you're much more sensitive to motion, head motion, than when you're measuring the connectivity of things that are close together. And it turns out that the younger babies, younger children, move more than older children. And so we're really not sure anymore about whether this early story of long-range connectivity is true. So what we would like to do, and what we wanted to do, is to tackle this, but not use resting state fMRI because it has this motion confound. So we used a different kind of method, and this is called diffusion MRI. And what this does, we're actually measuring the wiring of the brain, so the white matter that connects different brain regions together. And it actually, the reason it's called diffusion MRI is it's based on water diffusion, the fact that water's kind of jiggling around all the time, and it jiggles more along the axons than it does across the axons. We can measure, run a different kind of program with the MRI, we can measure that. And we can get these, you'd run tractography, and this is an image of some of the tracts in the brain. So what we wanted to use it for was to look for this long-range connectivity. We wanted to look for something interesting, some sort of meaningful connectivity. So we focused on what are called category-selective visual regions. So in the adult brain, we know that there are different regions that respond to different visual categories. So you have a region that responds to faces. You have a region that responds to places. You have a region that responds to tools. And it's been shown in adults that these different regions actually have different patterns of connectivity with the rest of the brain. And this kind of makes sense because we think they're encoding associations with those different categories. So faces, they're often making a sound, so perhaps they'll be connected to the auditory cortex. Tools perhaps will be connected to the motor cortex. So it's encoding those sorts of associations. A barking dog, the sound of a dog, and seeing the vision of a barking dog. What we wanted to ask in infants, really, the question we'd wanted to ask here was, are these connections that we see in the adult brain, are they a result of statistical learning? Is it because you've seen lots of dogs barking that your auditory cortex is connected to, your woof region is connected to your barking dog region, your vision region? Or could it be that some of these connections actually precede learning, and they might be shaping learning? So you might have some of this structure might already be there, and it might actually cause the selectivity, contribute to the selectivity. So let me just take you through this slide here. So the top panel here, the top brains, I'm just showing you the different colored patches. The red, the blue, and the green are the face, tool, and place regions, respectively. In the left column in the left hemisphere, the right column in the right hemisphere. The second row, what we've done is, in a group of adults, we're identifying the regions by their distinctive patterns of connectivity. I can tell you more about how we did that if you want later in questions. But so that's what you can see is we managed to recreate a lot of the pattern that this structure, these three different regions, we were able to find them just on their pattern of connectivity. We then go to the infants. This is 400 infants from the Developing Human Connectome project. That's a project run by King's College and Imperial. We're looking at 400 infants, term equivalent age. These are just newborn babies age. And you can see that, again, we can recreate this pattern of connectivity. They've got the same pattern of connectivity as the adults. So this is telling us that this subtle patterns of long-range connectivity are already present even in newborn infants. Okay, so what I've shown you so far is this executive control network. It's surprisingly mature. Long-range connections are present. But we still don't know that that means they're actually influencing learning. So we wanted to have a look at that. There's some very interesting work suggesting that a sort of adversity in young infancy, from many different sources, you get the same effects. Different adversities in young infancy can affect the development of this frontoparietal network. This suggests that it must be sufficiently well developed to be disrupted, but it doesn't tell us that it's influencing learning. So to look at that, what we did is we looked at a group of babies from the neonatal intensive care. This is an amazing medical success story. Neonatal intensive care has made tremendous progress in increasing the survival rates of young babies and increasing the health of these young babies, for example, babies born very prematurely. However, a significant number of babies still go on and develop impairments later in life. And at the moment, it's very difficult for neonatologists. The current behavioral and neural measures don't allow them to work out which particular babies are going to develop problems later in life. So we recruited a group of babies for functional MRI. We've got 65 babies here. And what we're going to do is we're going to look at different networks. So the way we did this is we took, for example, a network in an infant here, infant I. We're going to look at their network, and we're just going to say, how similar is it to a typical adult network? So these babies are scanned at term equivalent age. What we're then going to do is we're going to follow them up through the first year, and we're going to look at their learning. So in particular, we looked at motor learning here, because that's something that's easy to measure in the first year. So it's just clinical measures of how babies are learning to roll over, to crawl, to walk, and so on. What we wanted to do was let's see if disruption to the network at the age of birth can predict what happens in the development of motor skills later in the first year. What we found is, yes, it does. So first of all, not surprisingly, if you have disruption to your somatomotor network, that predicts that there'll be disruption to the development of motor skills. But surprisingly, if anything, even more strongly than this, development to this executive control network, the multiple demand network, is also predicting the development of motor skills in the first year. So what this is telling us is that in the same way as you or I, if we're learning to tango, we might be using our frontoparietal networks to learn those moves. Perhaps even young babies, when they're learning to crawl, when they're learning to walk, they might be using their frontoparietal network. Potentially this could be an intentional, perhaps a creative even, process that's going on. So just to give you a summary to sort of wrap everything up. So higher cognitive networks are surprisingly mature in infants from the age of term birth. We can see these subtle patterns of long-range connections in the brain. I've shown you that these higher cognitive networks seem to be facilitating learning even in the first months. And this is supporting the idea that infants may be much more active learners than we realized. And they may be potentially creatively generating models to explain what's out there and then testing them. This is the idea of, Alison Gopnik calls it the scientist in the crib. Just one final plug. We really need babies to participate in an experiment this week. If you have any babies, 6 to 24 months, or you know somebody that does, please send them our way. We'll give you €5 for 5 to 10 minutes online with your baby. Please just email me for the link. Thank you for listening.
That's a great pitch. That's for the people online as well, of course.
Great.
Not just in the audience. So thanks so much. I mean, it's very interesting to sort of pull at the threads, the connections, the creative connections through the four talks before we open up to questions, because it seems to me network connection is key. Network connection happens very, very early in life, it seems, and it would appear that the stronger the connections, the more creative thinking, the more creative people are. So Lorina spoke about the untapped potential in the brain. There appear to be dormant and perhaps inhibited networks that can be activated. Darya and Roger really helped us define what are the networks that map onto creativity and creative thinking. The default mode network, frontal networks, salience networks, and again, as I mentioned, the importance of coupling, strengthening the connections between networks as part of the creative process. And then Rhodri then pulled it all together in terms of how network connection occurs very early in life. But for me, I think the message and the sort of golden thread coming across all of the thoughts, and I think Darya alluded to this, was how we need to encourage creativity to build these network connections across life. And I guess in terms of our education system, how we're approaching education, I think there's some very important messages here in terms of trying to generate a creative thinking or creative approach to child development. because there's a brain science behind it, an involving brain science behind it. And I guess that's why we're here this week for Creative Brain Week. So we've got some time, and we may have some questions in the chat, or if we have questions here in the room. Feel free.
Hi. Thanks very much. This was fascinating, and I really respect that both the speakers in the room and then the speakers on Zoom were able to translate very complicated science to a layperson like myself articulately and clearly. So thank you for that. I have two questions. One for the professor in the center. It's kind of short. You did research with narratives, visual narratives in the form of a movie. Have you done any research on still images and how that, how people process still images in the sense of being stimulated by them? And my second question is kind of a follow-up. I'm a photographer and an artist. And I think there's kind of an axiomatic understanding that creativity is good. And I think it is, for sure. But my question is about the value of creativity in art specifically. So I'm wondering if anyone has ever tested if there's a coefficient of value in art as it relates to creativity. So for example, can you first measure the level of creativity that went into creating a piece of art? And then can you measure that level of creativity against other pieces of art? And then can you map that against what we consider value or valuable art? So for example, whenever I create photos, and I'm assuming there's other artists in here, there's a difference between me just kind of going through the motions and actually spending a lot of time to create something really creative. But I'm not sure other people can perceive that. And moreover, I'm not sure that the most creative stuff that I've produced is actually like worth it. Do you know what I mean? So this is kind of like a philosophical question, but I just wondered if you could maybe comment on that or riff on that question. Thank you.
So
So
here's a
we take
challenge for you.
stab.
There's a creative challenge for you. Perhaps the science question first.
Yeah, so yeah, thank you for your question. I mean, first of all, there's been a ton of research on still images, but I think, and that is sort of the more basic steps in cognitive neuroscience that we're building on when we're looking at this more holistic, naturalistic, evolving over time stimuli such as movies or short stories. But I think probably the thing that you're alluding to is that, or I imagine that in art, those still images sometimes have a narrative. So that's where I would go with it. That's where my mind is going, that there might be still images that are telling us a narrative that connects together. You know, whether that's an explicit narrative that everybody gets or it's an implicit narrative that we are co-creators in. And I would say that in that case, that there would be very significant you know, shared and coherent activity across different individuals. That would be a measure of the success of that narrative. And I suppose the proof of the pudding would be to test this empirically, but that would be my feeling, that in as much as you are transmitting a narrative, or one is transmitting a narrative with those independent, discrete pieces, that you are creating this wider experience, potentially transcendent experience, and so on. So that is, you know, in answer to your first question. And I will just say 2 cents with regards to your second question, and that is something that we're looking at now with regards to what happens in the brain when we're experiencing this, you know, very interesting, engaging narratives. Is that the one part that I shared is with regards to this common experience across different people. But what we find with very powerful narratives is that there's also subjective experiences that are very reliable, that are piggybacking on the same brain systems, and that are very strong, and there is an orchestrated interplay between those shared and intrinsic activities that are not shared but are very powerful. And, you know, again, I very tentatively would think that based on that work, that, you know, the stronger the artwork is, the more powerful those shared as well as intrinsic processes that are happening that we can measure in the brain. But I would stop short of the idea of how we put value or how we put a number on it, because we are not there yet. But that is my intuition as a neuroscientist, that you're really stimulating the brain. These networks are fully engaged and alive, and are creating sometimes very individual, intrinsic responses that maybe don't translate in that kind of agreed common denominator value of the piece, and maybe that's why sometimes you don't really know whether it's quote unquote worth it. But I think if we were to measure the brain, we'd find that it's very much alive.
So I mean, I just to add to that, I think there's a difference between the process of creativity and the engagement, the progress producing it, for you as an individual, as an artist, and then sometimes the value other people put on it. I mean, I'm just reminded of the work of Beckett, initially, I mean, his work was not valued, but over time, that changed. So that's how I would look at it. Rhodri, any comment?
Van Gogh too. Yeah.
Hmm?
Van Gogh too.
Yeah.
died in penury. I mean, something just on the still images. Going back to the still images, something that people do a lot is try to look at how using imaging to look at the response to images in the brain. They can look at what it is people are responding to in the thing and try to understand it. So, for are they merely representing the colors and the features, or are they getting some kind of deeper meaning out of it? And you can look at, say, the association between your response to different kinds of pictures, and try to get at those sorts of things. Something that we did, which suggests that neuroimaging can tell us something slightly counterintuitive, is we looked at people's imagination. We looked at the brain activity in response to people looking at images, and then the response to them imagining an image, and looked at the relationship between those things. And it was a little bit surprising. So you might imagine that there's a thing that goes right back to people like Francis Galton and so on, but you might imagine that when you imagine something, what are you imagining? Are you imagining the colors, the shape, the colors, the lines, or are you imagining something deeper? If you're imagining a flower, are you thinking about the smell and the beauty and these more abstract things? And you can ask people this stuff, but we looked at the brain activity, and what we found is that when people were imagining stuff, they were actually imagining them, the deeper properties, rather than the superficial characteristics. And what's more, people that reported clearer visual imagery, people that said they could see it more clearly, they were even more abstract. So they were, representing even more these sort of deeper things. So these are an example of, you can use these sorts of methods to start to unpack how people are thinking about things.
So,
yeah.
Question here, and then a question down here.
And we might engage some of our online audience as well. Is that okay?
Yeah, please. Yeah, online.
So one question from an online participant is, how do the cognitive studies of the brain networks embrace the lived experience of intuition, incubation, and what many artists call the unconscious?
Nice one.
That has to go to you as
yeah,
the unconscious,
okay.
I'm afraid.
So I suppose this is very much at the edge of science now in terms of what we can say about the unconscious. What we're trying to look at is how conscious processes of creativity, whether it's active or receptive creativity, are being expressed in the brain. And some of the things that's why we are moving away from, isolated brain region, isolated, individual, very artificial types of stimuli to look at that experience. And we bring individuals in to tell us what the experience is in a very holistic way with stimuli or narratives or other types that are not created for the laboratory, they're created for, an artistic experience. So I'd say we know some about what that lived experience is, and we know that it's actually very similar between different people when we have a powerful piece of art, but to the extent of the involvement of the unconsciousness in that, I would wait until the science catches up with that before I speculate, but that's just me.
Good answer. Maybe take the question front and then, okay. There's some in the middle and then down the front. Yeah, go ahead.
Hello. So I'm kind of interested in boredom and how important it is to allow yourself to be bored in the creative process. I think I saw Bob Dylan speak there a few years ago. He was interviewed about 60 Minutes, and he was asked about where the songs come from. he was reluctant to divulge. He didn't want to know. He was like, I don't know where they come from, and I don't want to know. I don't want to analyze it. You know, it's coming from the ether. You know, it's almost like being transmitted to him. That's the way he likes to see it. So that's kind of along the line of boredom as well. And sorry, just quickly, I suppose, on the children aspect, I was quite interested in the executive decision-making of children. Play, the importance of play. So there's a quote or something, we spend half our life when we're kids trying to grow up, rushing to be adults, and then we're adults, we spend a lot of our life as adults trying to be kids again. And we lose the skill of play. Because I think one of the speakers put up Ken Robinson's piece about education killing creativity. But I guess when I see children, and people talk about mindfulness, children are so in the moment, they're so in. And I suppose when they don't get bored, because they fill the space with play. And as adults, I think we lose that a lot. Sorry, that's kind of a lot of things. But yeah, so the two main points were boredom and I suppose play. So.
Do you want to? So Rhodri?
The boredom and indeed the unconscious experience as well a little bit. There is, I think, we are increasingly aware that there are some limitations of the way that we do psychology experiments, and that we always give people something to do and look at what is happening. But in fact, spontaneous thoughts that come out of nowhere, you know, most of, I think, is it 2/3 of in real life, 2/3 of the things we're ever thinking about are things that we've spontaneously occurred to us. And so there is an increasing attempt to study those sorts of things using psychology and neuroscience. Some of the speakers mentioned the field of mind wandering, which is this idea of, you know, what happens when you're not given something specific to do, when you're not on task, and I think relates to your boredom, you know, comes out of that kind of situation. It's richest when you're most bored. So there are attempts to study that and to look, for example, there are individual differences in mind wandering, or we looked at earworms, you know, those songs that just come into your head and go round and round, and it's actually quite a serious medical problem in some people. But you know, so that those sorts of things, and are people's brains different? We try to study that. So yeah, that. Yeah.
Did you want to make a comment? We have an expert here on creativity and play. Do you want to make a comment?
Yeah, or even just to expand on what you've said. So what we call boredom in neuroscience of creativity is incubation period. So, and a lot of research has been done on that with divergent thinking tests. So what they have looked at is they've looked at how the participants are given the divergent thinking tasks and then compared to participants who have been given time to just mind wander and those who haven't. And they have found links between creative production and incubation, but then the variables that come in then is the length of incubation time and individual differences, so it's kind of more nuanced, but it is being looked at, and it is something that affects creativity. And then with regards to playfulness, that's kind of my research area. And again, empirically, there's a researcher in Germany who has looked at this, and he's divided playfulness into 4 different facets. So one of them being other-directedness, lightheartedness, intellectual and whimsical. And what that does, what that questionnaire has kind of done in the playfulness research is it's allowed us to, I suppose, look at playfulness again in a more complex way and not just look at it one-dimensionally. And then again, I suppose how that relates to the neuroscience and how it relates to creative production.
So we've time for one last question. Thank you for that, by the way. So we want one last question here. Yeah.
This is really a question that struck me across quite a lot of presentations, which is, to what extent is there an issue or a problem to differentiate between creativity and art making? In particular, for example, we saw Saint-Victoire by Cézanne. By the time he's got to his 100th version of that, is there any creativity left? There's obviously art left.
Well, I mean, that goes back to how you define art, right? I suppose that would be a question for you. Where do you draw the difference between creativity and art?
Well, I would have thought it's more of a functional question in that if you want to, take the 100th Mont Saint Victoire and present it to a subject and say, okay, this is a creative work, but actually it isn't particularly. Maybe the first few were, but the 100th, is it or isn't it?
Well, I guess to my mind, I mean, for Suzanne, I guess it depends in terms of the process that he went through when he was doing it. Maybe it was more intricate, more challenging, maybe less challenging. Although perhaps what looks less impressive to us may actually have been more challenging to him toward the end. So, but the creativity. I mean, to my mind, it's not just what's produced. Actually, it's what you go through to produce it. And it's, and that's what the creative process, that's creativity is. I think that's the benefit of creativity. Okay, well, I think we're just spot on 1 o'clock, which is wonderful. Time for lunch. And just want to thank everybody, thank speakers who are here, Lorina and Rhodri, but also thank the pre-recordings. And I think it was a wonderful session. It was tightly coupled, very linked, very connected. So, and thank you for your participation.
Thank you.